that I found out yesterday.
Yep.
Okay, I will get started here. So my name is Kevin Lamp, and I did my master's research on bobcats in Western Maryland. And so I'm going to talk to you a little bit about that research today, specifically bobcat occupancy, abundance, and population genetics. So to begin, bobcats are medium-sized felid, native to North America. Females weigh approximately 6.8 kilograms, or right around 15 pounds, and males are a little larger, averaging around 9 kilograms and around 20 pounds. They're a generalist species capable of utilizing many habitats, as evidenced by their widespread distribution. As you can see in this figure here in the bottom right, bobcats, their range goes from southern Canada south through central Mexico. And then there are a couple locations throughout the United States where they don't exist currently. And this is along the East Coast here. And this is largely due to just the amount of human development, this high human population in this area, as well as this other area that goes along the southern edge of the Great Lakes and over into maybe southeastern South Dakota or so. And that's due to intensive agriculture. There's just not as much habitat available anymore, and so this area doesn't tend to have bobcats at the moment. Although some recent research was calling bobcats kind of on the edge of this range, and was seeing some dispersing individuals move into that area. So although bobcats are widespread, this does not mean that they're easily studied. So bobcats are solitary and typically not seen in groups unless it's breeding season or a female has kittens. Additionally, bobcats are highly mobile, moving multiple kilometers per day, and they can disperse distances of 160 kilometers or more, which is right around 100 miles or more. So this mobility that I'm talking about is on display in Maryland, where bobcats are being reported east of their known range. Little is known about bobcats currently in Maryland. There was a general predator survey conducted during 2015 through 2016, estimating bobcat density as 10.6 bobcats per 100 square kilometers. However, this survey wasn't designed specifically for bobcats. It was designed for coyotes, black bears, and bobcats. And it was in relation to a fawn survival study. So the lack of information and apparent expansion of bobcats eastward provided this opportunity to conduct research specific to bobcats in Maryland. So my main objectives were to, number one, assess population genetics, including population structure and genetic diversity. So population structure just refers to whether or not there are multiple distinct genetic clusters of individuals within an area. And that area is just defined by the researcher. So things that might cause population structure or different genetic groups would be barriers. So something as simple to think about as just a large interstate highway may cause bobcats on one side to be different than bobcats on another side. Another possible explanation for distinct genetic clusters would be isolation by distance. And so this would kind of refer to the dispersal or movement capabilities of the species in question. So in bobcats, you'd have to move pretty far to see isolation by distance. Just meaning that as you move from one location to another, the farther you go, the less likely that the genetics are going to be the same. And then to assess genetic diversity, that was one of the other goals here. And so I'll get into some of these terms a little bit later, but we were looking at things like heterozygosity and allelic richness at each loci or each locus within each population. So we needed to first figure out whether or not there were multiple distinct genetic clusters or populations, and then within each of those genetic clusters, we would see what the genetic diversity looked like. The second objective was to evaluate and predict occupancy across Western Maryland, and occupancy just refers to the distribution of bobcats. So if you think back to that image I showed of the United States, so that obviously is bobcat occurrence across the United States. We were just looking at bobcat occurrence in Western Maryland. And so we want to tie occurrence maybe to specific land cover covariates, so different land cover types, or maybe it's habitat type in particular, or some sort of environmental characteristic, could be elevation or ruggedness, those sorts of things, and how that affects bobcat occurrence. The third objective was to estimate density using 2 different methods and then compare the results. And so a comparison would help us determine which method would be most useful for Maryland's wildlife managers to monitor the species into the future, as well as it would offer information to other bobcat researchers and managers in other states. But the biggest question we have right now, before we get into the research here, is how do we complete these objectives? How do I get the answers I'm looking for if bobcats are so difficult to detect, like I mentioned earlier? And so the answer is non-invasive survey techniques. Non-invasive survey techniques are non-intrusive and they cause little or no disturbance. So instead of capturing an individual and putting a collar on it, or capturing an individual to take some genetic sample, we wouldn't need to do that. And so in particular, some examples would include camera trapping. So you don't need to actually handle the animal for collecting data via cameras, or DNA collection via hair scatter environmental DNA. And so in particular, like hair snaring, for example, it would use a device that an animal would come in contact with, and then that device would grab and pull hair out of the animal. So it's kind of a passive type thing. The animal's got to come in contact and leave hair behind. Scat surveys are a little bit more active. It usually refers to people hiking a transect and then just searching for scats, and just looking on a specific section of trail or something. Or you could use dogs. There's scat detection dogs, well-trained dogs that search out scat, that make it much easier to find scat in a along a transect, especially if it's not a trail or a road or something like that. And then environmental DNA, just to clarify what that means, environmental DNA just refers to DNA that's maybe left in the environment, but not necessarily visible to the naked eye. So one option that we had considered when conducting this research was collecting environmental DNA from snow tracking. So I was doing snow tracking to collect hair samples at one point, but we were also thinking that maybe we could scoop the snow around a track, and because an animal walked through there, you're leaving skin cells behind, those sorts of things. And so just collecting the snow, melting it down, and then analyzing that water for bobcat DNA was a possibility. A lot of times this is done in like streams for aquatic species. You sample the water in the stream, and then you can get an idea of what species are using that stream. So in Maryland, I mentioned that previous study, cameras had been used for that study. However, there hadn't been any genetic methods used for bobcats. The state had done some genetic sampling for black bears, I believe, in the past, but they haven't done anything with bobcats. And so we need to figure out which methods would work, since a large portion of my research depended on these genetic methods. So I conducted a pilot study to determine the most effective genetic methods to detect bobcats, and then to identify individuals via their genetics. So my study area where I conducted all my research was in Western Maryland, an area that's characterized by forest cover. Garrett County lies to the west, and it's on a rugged plateau, whereas Allegheny County is within this ridge and valley landscape. I selected 3 study areas corresponding to the land within and adjacent to Potomac Garrett State Forest, Savage River State Forest, and Green Ridge State Forest. And if you look here in the figure, Potomac Garrett State Forest is in that red color, Savage River is in the blue, and Green Ridge is in the green. So to evaluate non-invasive genetic survey methods, I conducted a pilot study from January through March of 2019, in which I completed a hair snaring survey that included 6,840 trap nights, a scat sampling survey that included surveying 318 kilometers of transect, and a snow tracking survey in which I would collect hair from tracks or natural snags. And this survey included 626 kilometers of transect. So just to explain these photos a little bit, the image on the left side is of the hair snare that I used. It's a cubby style hair snare. I believe this was originally designed for Fisher, and then it was redesigned for bobcats by a PhD student who did some work in West Virginia. And so I'd spoken with this PhD student, and I had added a couple things to the cubby, but the basic concept here is that, you put bait and lure inside, and the bobcat would want to enter to investigate that. And there's bore brushes, if you look close, used for cleaning the bores of guns. And so it's a wire brush, right? And as the bobcat goes inside to get at the bait or the lure, it would pull hair out and then leave it behind. And so that's the idea behind the hair snare. If you look at the figure or the image in the top right, that's just me sampling a piece of scat. And while that seems pretty self-explanatory, there's some important aspects to sampling scat. So just a scat laying on the ground, there's a specific spot that you'd want to sample from. And so in this image, you can see I'm taking a sample from the side of the scat. And that's because as soon as that scat is deposited, it's in contact with the ground on the bottom. And so there's bacteria and fungus and just the moisture that's degrading the DNA from the bottom, whereas the sun is degrading DNA from the top. And so if you sample from the side, it generally has a slightly higher chance that you'll be able to get a genotype out of that DNA when it comes to the analysis. The image on the bottom right is just a bobcat track in the snow. And so when I said I did snow tracking surveys, we would hike a transect, identify a track line as bobcat track line, and then follow that track line, usually backwards, just to make sure that we weren't necessarily stressing the animal out by pushing it if it was a fresh track line. And then as we followed it, we'd look for hair. So you just get down, look for hair in the tracks, around the tracks. If the bobcat went underneath barbed wire, you might look to see if any hair was left behind there, or on natural snags, like I mentioned, it could be greenbrier, the thorns. I actually found some hair on some greenbrier one time, or maybe a rocky outcropping that the bobcat is walking along. And so it's just searching for hair along the track line. So the results showed that each of these methods gave few bobcat detections, which wasn't ideal. And in addition, there were few individuals identified. But if you look closely, scat sampling shows a little bit of promise. So I collected 48 predator scats, and of those predator scats, 9 were identified as originating from bobcats. And of those 9, 5 gave us a genotype, or we were able to identify the genetic code for that individual at the specific locations on the chromosome that we're looking at. And so with that, you can see that, sure, it's a small sample size, but a little over half of the samples that were from bobcats gave us this individual identification. And so armed with that information, I moved forward with scat sampling, knowing that I needed to increase my sample size. So to just go back, we're going to start out with the first objective, assessing population genetics. And as I mentioned, I needed to increase sample size, so I designed a scat sampling survey from May through August of 2019. The survey covered 11 weeks, and with the aid of 3 technicians, I surveyed hiking trails, off-road vehicle trails, and closed roads to sample predator scat. So if we look at the figure here on the right, you can see each study area, and then you can see all the black lines within each study area, and those correspond to all the transects that we hiked. The technicians and I surveyed 488 kilometers of transect on 5 separate occasions. And so multiply 488 by 5, and that's approximately what mostly just 3 people hiked that summer, but 4 of us in total. We collected 914 scat samples, and then I went back and removed some scat samples that were from low quality or presumed black bear scats. Now we had images to go back and look at, so I was able to sift through the images and really make sure that the samples we were sending to the lab would give us, a good chance at getting DNA. I didn't want to, waste a lot of money, being that the analysis is expensive. So this gave us a total of 816 stat samples that we sent to the University of Idaho to be analyzed first to the species level and then to the individual level. So the species identification was done using mitochondrial DNA. And of the 816 scat samples that I sent in, 241 were identified as bobcat scats. And the individual identification was done using 10 microsatellite loci. And of the 241 samples, 83 gave a consensus genotype. And so we run these samples multiple times through this analysis, through this process, just to make sure that the genotype we're getting is the true genotype. So that's what I mean when I'm talking about a consensus genotype. Of those 83 samples, there were 35 individuals, and 12 individuals were captured once or detected once. 23 individuals were captured or detected more than once. So to analyze my genetic data set for population structure, I used a program called Structure and a principal coordinates analysis. So Structure assists in determining the number of distinct genetic clusters I was talking about before. So what we would call a population within my set of sampled individuals. And then a principal coordinates analysis just helps visualize any clustering. So they're both doing kind of the same thing, whereas Structure is looking at the genetics themselves to figure out, okay, how similar are these genetics? Where principal coordinates analysis is taking into account, any variation, whether it be, distances or where the sample was collected from. So like my study areas and things like that. So we'll see the results here in a second. And then for each population, I calculated the number of alleles per locus and the heterozygosity to evaluate genetic diversity. So to clarify some of these terms that I've been talking about so far, might take you back to biology class a little bit. A locus is just a specific location on a chromosome. And so if you look at the figure in the bottom left, the location, so this is just one chromosome, the red and the blue, it's just split apart. But if you look at one location, so if you took a vertical line and drew right through it, that would be a locus, a specific location on that chromosome. So if you see it says gene loci, it's pointing to three different loci. If you look at one of those, that would be called a locus. The alleles are alternative forms of a gene found at the same locus. And so if you're looking at that figure in the bottom left again, the alleles would be the capital P on the red piece of the chromosome and a capital P on the blue piece of the chromosome. So in that case, if you look down at the bottom of the figure, it says homozygous. And so that's just saying that has the same allele twice. Whereas if you look in the far right of that figure, it has a capital B and a lowercase b that has two different alleles at that particular locus. Another way to look at this would be to look at the Punnett square. And you can see from the Punnett square here, which is the A's and the B's and the Z's, you can see that there's an A and a Z on the left, and that's referring to one individual at a particular locus. If you look at the A and the B above the grid, that's another individual at the same locus. You're looking at the two alleles at a particular locus for these two individuals. And so if these two individuals were to have an offspring or multiple offspring, the possible combinations of these alleles at this particular locus or their offspring are shown in the grid. And so you can see that there's the cell in the upper left has two A's. This would indicate a homozygous individual, whereas the other three cells would be considered heterozygous individuals at this locus because they have two different alleles at that same locus. And so when I refer to heterozygosity, I'm referring to whether or not an individual was heterozygous or homozygous. In particular, we're talking about the proportion of individuals that are heterozygous when I refer to heterozygosity. So I'll explain that a little bit more with the results. So the plot on the left shows the output from the program Structure. The points in the figure just represent the mean log likelihood values at 1, 2, 3, or 4 possible populations or possible genetic clusters. As you can see on the x-axis, there's 1, 2, 3, or 4 possible genetic clusters. And all this figure is saying is that the possible population with the greatest likelihood is 1. And so this is just basically saying that within my genetic data set, it seems as though all of these individuals are coming from the same genetic cluster, the same genetic population. Oh, let me go back here. The principal coordinates analysis on the right, this just shows each individual. So each point within this plot is one of the 35 individuals, and they're color coded based on the study area that they were found in. And so what I'd be looking for in this plot is if there was any clustering. If there was clustering, maybe if you want to look at those red diamonds close to the center, you can see there's about 5 of them there. You could maybe make a case that there's a little bit of a cluster there, but it's not enough to really say anything definitively. Where if you look across the whole plot, all of these individuals from all 3 of these study areas are kind of just mixed together. And so this just gives more evidence to say that we have one genetic population in Western Maryland. So as far as genetic diversity goes, bobcats in Western Maryland are genetically diverse. If we look at columns 2, 3, and 4 in this table, you can see that we've got 3 different labels. We got an A, an HO, and an HE. The A indicates the number of alleles per locus, and the higher the number, the greater the genetic variation at that particular locus. And so if we look at the average, you can see that the average is 7.1 alleles per locus, which is good. If we were to see a specific locus that had 1 allele, that would be very bad, because that would indicate that throughout the entire population or throughout your entire genetic data set, all the individuals had the same exact allele at that specific locus, and they would be homozygous, so you have the same allele twice at that locus. And so that'd be a bad thing, but we don't see that. We have a pretty good average there. If we look at the 2 columns to the right, the HO and the HE, they just refer to heterozygosity. And so when I said heterozygosity, I'm referring to the proportion of individuals that are heterozygous within the population. And so if you want to think of these as percentages, you could think of it as that. So if we look at the first locus, F124, you can see that like 85% of the individuals are heterozygous at that locus, meaning they have 2 different alleles at that locus. And based on the most basic population genetic model, where individuals are randomly mating, there's a large population size, we would have expected the allele frequencies to show us an expected heterozygosity of 0.754 there for that first locus. And so you can see that there's an excess of heterozygotes, which means there's an excess of genetic variation, basically. So if we look down at the averages, the same thing is shown down there. We have this excess of heterozygotes in the population, meaning that there's plenty of genetic variation. So this lack of population structure was not surprising, since the region I was able to sample was relatively small in comparison to bobcat movement. So I mentioned at the beginning, bobcats can disperse 100 miles. And so the area I was sampling was relatively small in comparison to the mobility of the species I was studying. In addition, there's no obvious barriers to gene flow in Western Maryland. If you think about Western Maryland, yeah, it's rugged. There is a couple major roads that run through there, but nothing that's so large that a bobcat couldn't cross it. If you think about maybe the Potomac River, sure, cats generally, you think of them as not wanting to swim, but bobcats will swim, and it's not that large of a river that they wouldn't cross. And so it's not crazy to think that bobcats are able to move across that as well. However, as I mentioned in the beginning, as bobcats expand to the east, throughout, in the future here, there's going to be more barriers that they come across. So there's large interstate highways, there's areas of intense human development right around Baltimore and Washington, D.C. And then further east yet, the Chesapeake Bay is a large barrier for them getting onto the Delmarva Peninsula. And so there are some barriers that they're going to have to work around as they move east. And then lastly, I had a small sample size of 35 individuals, and that could impact the results. Most of these metrics that I used are robust to small sample sizes, but with a larger sample size, I may have detected more alleles in the population, which would just go to show you that there would be even more genetic variation. So it wouldn't necessarily change my results in saying that bobcats are genetically diverse, but it would show you, how many alleles are truly in the population. So now that we have completed the first objective, we're going to move on to the second objective, which is to evaluate and predict occupancy across Western Maryland. So to collect detection data to estimate occupancy, I conducted a camera trapping survey over 8 weeks from January through March of 2019 and 2020. The survey included 120 cameras, 40 in each study area. So if you look at the figure on the right, you can see each of those grids contains 40 cells. So there'd be one camera in each of those cells. Each cell was around 5.5 square kilometers in size. And we placed the cameras within those cells at a place that we figured would offer the best chance at detecting a bobcat if one was there. So we prioritized early successional habitats, travel corridors, maybe ridge tops or valley bottoms, game trails, hiking trails, those sorts of things, as well as edge features. So it might be the change in habitat from a mature forest to a timber cut. It could be agriculture to a forest edge, just any sort of edge feature. And then we also considered accessibility to some of these sites because this is some of these areas are fairly remote. We had to think about, how we were going to access, how much time it was going to take, in order to get to all of these sites once a week to check them. And so we considered this accessibility and the overall spread of cameras to make sure that we could get to all of them and we were minimizing unsampled areas. Volunteers and I visited cameras weekly to maintain the cameras and then replace the baits as well. So in 2019, I collected almost 125,000 images. And of these images, I documented 105 weekly bobcat detections at 51 of 120 camera locations. And in 2020, I collected 89,000 images, or right around 89,000 images, offering 78 weekly bobcat detections at 42 of 119 camera locations. And so the figure on the right, you can kind of see where those detections took place. So if we look at the Potomac-Garrett grid on the far left, you can see that the whole southern portion, we didn't detect bobcats in either year. The black cells indicate detections, bobcat detections that we had in both 2019 and 2020. The lightest gray color, or there's two cells in the northwest corner there next to each other of Potomac-Garrett that have that light gray color, indicating detections only in 2019, and then that dark gray color would indicate detections that only occurred in 2020. So I organized my detection data into 8 7-day sampling periods. So I mentioned creating weekly bobcat detections, and so weekly detections would just mean that, if we had this camera running for 1 week, and on Monday we detected a bobcat, and we detected another bobcat on Wednesday and another one on Thursday, it wouldn't matter that we detected those 2 bobcats on Wednesday and Thursday. As soon as we detected one, we'd consider that cell or that camera site occupied for that 7-day period. I calculated covariates to help explain bobcat occupancy by buffering the camera locations by a 1.85 kilometer radius, which equated to the estimated female bobcat home range size of around 10.75 square kilometers. So after creating this buffer on the map, then I extracted data on forest cover. If you look at the occupancy covariates in this table, I extracted data for forest cover, early successional cover, road density, stream density, and terrain ruggedness. All, for each camera, I did this just within the buffer. And then using the covariates, I modeled bobcat occupancy by first determining the top detection model from a set of 4 models while holding occupancy constant. And so the reason that we need to do detection first is because we have to first account for detection at a site, because there are 3 possible outcomes of our surveys. So the first outcome is that bobcats do not occupy the grid cell, and therefore we could not detect them on camera. They're just not there. You're never going to get a picture of them. The second option is that bobcats occupy the grid cell and were detected on camera. So they occur in that area, and we did get one on camera. Or the third option is that bobcats occupy the grid cell, but we did not detect them on camera. And so that's what we need to account for, this imperfect detection, the imperfect ability to detect bobcats. We're not going to detect them every time that they're there. And so we need to make sure that we account for this probability of detection. And so that's what the determining this top detection model is going to do first. We figure out what's the best covariates to explain why we were detecting them. And then from that, we can estimate occupancy. So after I did the detection model, I paired that top detection model with a set of 25 occupancy models with various combinations of the covariates that you see, the occupancy covariates you see on the right. The top detection model included only lure type. And so of the two lure types I used, beaver castor and catnip oil. Beaver castor improved the probability of detection over catnip oil. However, the confidence intervals for the probability of detection overlap. So there's not a great difference between the two, but there's a little bit of difference. Statistically, it doesn't really exist, though. It doesn't, it's not a significant statistical effect. The top occupancy model included road density, percent early successional cover, and stream density as negative predictors, as you can see in red, and terrain ruggedness as the only positive predictor, seen in black. And lastly, I averaged the top 7 models to create a model that would predict occupancy across the 4 westernmost counties in Maryland. Just before I move to the next slide, it's important to note that each of these top 7 models includes road density and terrain ruggedness. Just kind of goes to show how important those 2 covariates were in explaining Bobcat occurrence. So from left to right, or west to east, we have Garrett County, Allegheny County, Washington County, and Frederick County. Each hexagon in this figure is color-coded to represent the probability of occupancy. The lighter the color, the lower the probability of occupancy. The darker the color, the greater the probability of occupancy. And so if we look at a specific area in Garrett County, there's this really dark area kind of on the east side of Garrett County. And that corresponds pretty well to Savage River State Forest, which is one of the most rugged areas in Western Maryland and has few roads. So as I mentioned, those 2 important covariates, terrain ruggedness was a positive predictor, meaning that with more rugged terrain, you're going to see the higher probability of bobcats occupying that site, as well as road density, which was a negative predictor, meaning that as you see an increase in road density, you see a decrease in the probability of bobcats occupying that area. So that seems to make sense. And if you look to the southwest of that really dark area, you see a really light area in Garrett County. And so that corresponds to an area kind of around Oakland, Maryland. There's a little more agriculture occurring in this area, definitely more roads, it's less rugged. And so that, that goes to show you that this model is picking up on those things and indicating that this area would have a lower probability of bobcats occupying. So as I mentioned, the most important covariates to bobcat occupancy in Western Maryland that I was examining were terrain ruggedness and road density. As I mentioned, terrain ruggedness, with an increase in terrain ruggedness, you saw an increase in bobcat occupancy, and with an increase in road density, you'd see a decrease in bobcat occupancy. However, there are some temporal limitations to my study. So my covariates may impact bobcat occurrence differently throughout the year. I did my research during the winter. And so what I'm getting at is during the summer or other months, these covariates may impact the bobcat occurrence differently. So in the winter, rugged terrain may be more important for bobcats that are trying to move through this landscape undetected. During this time of year, there's no vegetation or maybe lower vegetation, and so there's less cover to hide their movements, and so they may be using rugged terrain to move through the landscape undetected, to hunt for their prey and things. Additionally, the study is done during bobcat breeding season, which is going to increase bobcat movement as they look for breeding opportunities, and this could increase detection rates, as well as during this time, you're probably going to have a few more dispersing individuals on the landscape, and that may artificially increase occupancy. You may have these dispersing individuals. I don't want to say necessarily lower quality habitat, but habitat that wouldn't otherwise be occupied. They may be there, and so they may show up on our camera, and that would tell our models that, hey, this these habitats in this area, may positively increase, might increase bobcat occupancy, be a positive predictor when in reality it's not. And so there's some limitations, some caveats there. And lastly, the predictive model may not be well suited for Washington and Frederick counties. We wanted to extend our estimate a little bit to the east, but Garrett County and Allegheny County are much different than Washington and Frederick counties, especially eastern Washington County and most of Frederick County. Frederick County and eastern Washington County are much less rugged. There's much more roads, much more agriculture, and so my models may not represent the areas in Washington and the Frederick counties as well as they could. But you look at western Washington County, there's still some rugged terrain there, a little less agriculture, and so we wanted to extend this out a little bit, being that bobcats are moving to the east. So now that we've completed my second objective, we're going to switch gears and move to my third objective. Estimate density using 2 different methods and then compare those estimates.
Hey, Kevin, quick
Yep.
question from Chris. Does the one hexagon in southeast Frederick County with elevated occupancy, is that Sugarloaf Mountain?
Where are we talking about?
Is the one hexagon in southeast Frederick County, I guess it's just the one that down here, the darky red one.
I'm not sure. I'm not exactly sure. I'd have to look at that. I didn't look at that too close. I mean, I looked at a lot of the cells along the border, or the hexagons on the border between Washington and Frederick counties. And there's clearly some more rugged terrain there. And I want to say, there's a lot more forest cover in that area, although forest wasn't an important predictor in my models. I would imagine that in an area that has a much less forest cover, that forest would be much more important. And so I'm not sure exactly what that hexagon is picking up on, although I could pull that up on like Google Earth or something and take a look and see what I think.
Okay, thanks.
Okay, so my first method to estimate bobcat density was camera trapping. So as in the occupancy methods, I organized the detection and non-detection data from the camera data into 8 7-day sampling periods. I used the same covariates as I did in the occupancy analysis. However, I used Royal Nichols models to estimate bobcat abundance using the same model set strategy. So instead of using occupancy models, I used what's called a Royal Nichols model, which is very similar, although it's estimating abundance rather than the probability of occupancy. So again, I modeled detection first while holding abundance constant, and then use the top detection model in each of the Royal Nichols models to estimate abundance. Again, the top detection model included lure type, with beavercaster improving the probability of detection over catnip oil, although it shows even less of a difference through the Royal Nichols models, through the abundance modeling. Just kind of goes to show that lure type didn't add that much to explain why we were detecting bobcats. The top Royal Nichols model included road density, percent early successional cover, stream density as negative predictors, and then terrain ruggedness as the only positive predictor. So this was the same as the top model in my occupancy models as well. And so I used this model to predict bobcat abundance across Garrett and Allegheny counties using the top model. And the figure on the right shows abundance per hexagon. So the darker the hexagon, the greater the abundance. The total abundance of all these hexagons is 162 bobcats. And to calculate the density, I divide the total abundance by the area encompassed by all these hexagons. And the density estimate was right around 6 bobcats per 100 square kilometers. So again, you can see that this dark area in eastern Garrett County just that centers pretty closely over Savage River State Forest. Like I said, it's very rugged, very remote, few roads. And then if you kind of look to the east, just into Allegheny County, you can see there's a ridge. I believe there's a ridge that runs through there, and so that area has a lot more rugged terrain, and so the model is picking up on that as well as over there in Green Ridge State Forest in far eastern Allegheny County. The second method I used to estimate bobcat density was scat sampling. So the scat surveys during May through August of 2019, these were the surveys I used to collect genetic data for the population genetics analysis in objective number 1. And just to remind you, my technicians and I surveyed 488 kilometers of transect on 5 separate occasions. We collected 816 scats, of which 241 were originating from bobcat. And of those 241, 83 gave a consensus genotype. And of those 83, there were 35 individuals. 12 were captured once and 23 captured more than once. I organized my transects into half-kilometer segments and then assigned, each segment center represented a trap, and then each scat was assigned to the nearest trap. So each scat was assigned to this half-kilometer segment in a specific location, and that's just to format the data for the models I was going to use. And so I modeled density using 8 spatially explicit capture-recapture models. I used observer, which is just the person who collected the scat, and Julian date, which is just the day of the study. So the number of days we collected scat. So the first day we were out surveying transect was day 1, and the last day we were out surveying transect was day 81. And then I used sex as a covariate on this parameter called sigma. And sigma just estimates how far from an individual's activity center and that individual can be detected. So it's nothing, it seems, when I talk about parameters or we're talking about this specific parameter, it's not that hard to think about. It's just, the baseline detection that I talk about above with observer and Julian date is just, our ability to detect them, just like we were talking about in the occupancy and the Royal-Nichols models. And sigma is telling us, okay, how far can you go from an individual's activity center or its home range center before you cannot detect them anymore. And so we figured sex would explain that, because male bobcats tend to have larger home ranges than females. Usually males overlap about 3 to 4 female bobcat home ranges. The top spatially explicit capture-recapture model was the null model, which just indicates that observer and Julian date have no effect on our baseline detection. And the rate of decay of detection from an activity center, sigma, did not vary by sex, which was a little surprising. The top model estimated bobcat density is 4.14 bobcats per 100 square kilometers. It's a little bit lower than the Royal-Nichols model. And to compare these density estimates side by side, we can see that Royal-Nichols estimate was slightly larger than the spatially explicit estimate. And if we look at the 95% confidence intervals, you can see that there's some overlap. And so these estimates may not differ as much as just looking at the point estimate itself. There are some differences in the study design, though. So the Royal-Nichols estimate was created from the camera trapping data on public and private lands. And so we used land cover covariates because we had some variation. We had some areas that were, I don't want to say completely agricultural, but much more agricultural. Then we had some areas that were mostly forested, some areas in between. We also had, much more variation in road density and those sorts of things, because some areas were on private lands and some areas were in the middle of Savage River State Forest, which doesn't have any roads. And so we had the variation in order to include those covariates to help explain abundance, whereas in the spatially explicit estimates, it was on public lands completely. And so there wasn't a lot of variation. It was mostly forest covered. So we weren't able to try to explain density based on these different land cover types. And then, as well as we had these differences in the timing, right? The Royal-Nichols estimate was done from data in the winter, where you have these dispersing individuals that might be increasing your abundance estimate. Breeding season is going to help increase detection. Whereas in the summer, not quite as much. You don't have those dispersers on the landscape, or at least not as many. And then, it's not breeding season. Females are with kittens, and so you're not necessarily going to have this really high detection rate that you might see during the winter. So the two methods that I used to calculate bobcat density offered similar estimates, indicating that detection and non-detection data paired with an appropriate study design can be useful for population estimation of a solitary species like the bobcat. The consensus in the literature is that males have larger home ranges than females, but my top model suggested that sigma, that parameter, estimating your ability to detect them the farther you get from their home range or the center of their home range, it didn't seem to vary by sex. And so after looking back at my data, the lack of data may explain this, because on average, males were captured 1.86 times or detected 1.86 times, and females were detected about 2 times a piece. And so without, without an adequate number of recaptures, I shouldn't say adequate, without more recaptures, it was hard to show, any variation in that sigma parameter. And so, it wasn't going to split them apart and say that there was this difference between the sexes. My spatially explicit density estimate is the lowest density estimate in the Central Appalachians. And so this could just be due to low prey density. So other studies have mentioned that, these mature forests have lower prey density. And so, that just feeds up the food chain. And so you get bobcats that, or fewer bobcats because there's just fewer prey species or less food on the landscape for them. So in kind of conclusion here, with this objective, we're trying to figure out what methods were useful for population monitoring in Maryland. And so is scat sampling the answer? It provided, more precise estimates, but the genetic analysis is very costly, and it can be time consuming. The camera trapping, it provides less precise estimates, and cameras have this high initial cost, but you can use them year after year, and you can monitor multiple species at the same time. And so there's, there's a benefit to that as well. In my opinion, I would recommend a combination of both these methods. So, cameras, spending the money on cameras, like I said, it's going to be costly up front. But if you use them year after year, and if you're looking at detecting the trend in the bobcat population, so it's more of an index. If you use it like that, you can follow the population through time, with high resolution if you're doing it every year, even every 2 years. And then every so often you come back and you do a scat sampling survey, maybe once every 5 years or once every 6 or 7 years, something like that, just to obtain that precise population estimate. So in conclusion, the takeaways of my research are that bobcats in Western Maryland belong to a single genetically diverse population. Areas of greater terrain ruggedness display greater probability of bobcat occupancy and a higher abundance. Areas of greater road density may limit bobcat occurrence as well as abundance. In detection, non-detection data from the camera trapping may be useful to estimate density. And just in general, bobcat density seems to be a little bit lower in this area than some of the other areas nearby. So I'd like to acknowledge my funding source, Maryland Department of Natural Resources. They were able to come up with the money, as well as come up with the money themselves, and then pull from the federal Pittman-Robertson money, the Wildlife Restoration Act, to support my research, as well as some money from the USDA. I'd like to thank my committee members, in particular my advisor, Dr. Jacob Bowman, and then tons of people that worked on my project. Various people within the Maryland Department of Natural Resources, state park staff that helped me out, gave me a place to stay while I worked out there, various landowners, various researchers at different universities across the United States, 11 technicians, other graduate students, as well as family and friends. And with that, I will take any questions.
Thank you, Kevin. This was great. Can we unshare and come back together for questions as a group? All right. If you have a question for Kevin on bobcats and his research, you can put it in the chat box or raise your hand and I will acknowledge you, and you could unmute and ask him yourself. Earlier on, we did have Melinda asked, how do you measure or characterize terrain ruggedness as a covariant?
So that was a tough one. We found some data. Well, I shouldn't say we found some data. We used data on elevation, and so using some geographic information systems, we had like a layer. If you want to think of like an image that has pixels, right, across the whole landscape, has these pixels, and each pixel would indicate a specific elevation. And I believe our resolution was pretty high for this. I believe it was 1 meter resolution, meaning that each pixel across all or both of these counties was 1 meter square. And so for each meter pixel, we had an elevation for that pixel. And so we basically pulled that layer apart into slope and aspect. And so basically our terrain ruggedness layer incorporated slope and aspect to get at ruggedness. So it wasn't just the change in elevation, it was okay, the change in elevation as well as which direction the slope was for each individual pixel, each meter by meter pixel, all the way across those two counties. And so we use that layer to calculate this I'm trying to remember exactly, I think it's called a vector ruggedness measure. We got a value, basically a ruggedness value for each one of those pixels. And then, like I said, we at each camera, we had that buffer that we placed around it, just a radius of 1.85 kilometers. And we pulled out the ruggedness for that buffer for each camera, so all 120. And then we averaged the ruggedness value, which wasn't ideal, but we couldn't think of a really good way to include all of those ruggedness pixels, right? Because we're talking a very large area, 10, almost 11 square kilometer area, and we've got data for each meter by each square meter. So we ended up averaging it, but it still came out as important. I imagine if we were to look, find a better way to show that variation across that whole buffer, that maybe it would have been even more important.
Sam asked, and kind of going on with what Sam asked, is when you said that you had your camera data and got hundreds of thousands of images, Sam wants to know if there are any unexpected species captured by the cameras. And I wanted to know, how do you go through such vast amounts of data to find only the animals that you're looking for?
So both of those go hand in hand, right? I ended up hiring 5 undergraduates at the University of Delaware who used a program called Time Lapse. And so we sent them the whole data set for a specific camera, one at a time. And they had to flip through every photo on their computer. And this program then allows them to, I created what's called a template, has all the different species that could occur. I think I lumped some species, like if the camera is being set off by small birds or something like that, or small mammals. I think I just have a category for all small mammals or something like that. But they had to go through each photo, and they would click on it and say, they would click on a category. So say they did see a bobcat, they'd click on the bobcat in the template, and then they'd have to click on the photo where the bobcat was. It marks the photo as having a bobcat in it, and if there was 2, you could mark both of them, so then it will record that there's 2 bobcats in the photo. So they did that for every single photo, and then at the end it spits out a table, and it says, in this photo we had a bobcat, or in this photo we had a wild turkey. And so it took a lot of time. I'm sure a lot of those undergraduates would tell you that they spent hours and hours on it, but to some degree it's exciting because you don't know what's going to pop up next. And so when you talk about any unexpected species, there was a woodcock on one of our cameras, like right in front of the camera. I think one of our cameras was near a stream, and so a beaver popped up onto the camera. We had a couple owls, I think, as well. But most of the images we had were, what we were looking for is, mesocarnivores, coyotes, bobcats, red fox, gray fox, fishers, those sorts of things.
No Bigfoot.
Nope, no Bigfoot.
Okay. So let's see. We have Maddie asked, "What's your favorite thing you've learned about bobcats in your work, research, and schooling that you want others to know?"
I guess don't feel bad that you haven't maybe seen one, because I worked out there for 3 years and didn't see a bobcat in my study area. They're, like I mentioned, they're solitary, they're secretive, they know how to move through the landscape and not be seen. And so if you're trying to see one and you still haven't seen one yet, even somebody who spends time looking for them hasn't seen one. I saw one here in Pennsylvania driving. I think I was driving around at midnight, coming back from helping with some deer surveys, and I saw one up in north central Pennsylvania run across the road. But yeah, during my whole master's, I didn't see a bobcat.
Well, that lends me to, when you were saying you were collecting the scat, and you collected the scat. If you were collecting all the scat, not all of it, it was all predator scat. So does it look similar to other things? And if so, what does the scat of the bobcat look similar to that it was so confusing?
Well, it's there's been some studies done on this in the past. So there's been a lot of studies that look at maybe survival of a prey species of bobcats. So if you think of whitetail fawns, and when they find a dead fawn, they may find scat nearby, and so they attribute, in the past anyway, they were attributing the fawn's death to predation, and they would attribute it to a specific scat. And a lot of times they would just use morphometrics. They just look at the scat, the characteristics of the scat, and say it was from a coyote or it was from a bobcat. More recently, there's been some genetic studies looking at those characteristics that a lot of these scat and track books are telling you, and it's not as cut and dry, as black and white as you'd expect, or as the book would make it seem. And so we actually, an undergrad for their senior thesis did basically that. They looked at the scats. They had, I think, 5, because I took a picture of every scat that we collected before we sampled it, and they had, I think, 5 volunteers classify every scat that we had collected as coming from some species based on some of these categories that these scat track books tell you. So like some of them for bobcats are, if it doesn't have tapered end, it's a blunt end, if it's segmented, those sorts of things are kind of attributed to bobcats. Well, come to find out, about 60% of the time those are correct, and 40% of the time they're not. And so basically it's really hard to identify scats just by looking at them, is what it comes to. Now, like I said, there's some general characteristics, but I mean, if you're wrong 40% of the time using those characteristics, it's not that great. And so I guess, one of the most important things I learned from this was just that, the fact that you've got to do the genetics to really know if the scat's coming from the species you think it is.
Interesting. Jean asked, do bobcats dig up prey? Is their hunting stereotyped?
I wouldn't say they generally dig up prey, but they'll cache prey. They'll kill something and cover it up and come back to feed on it later. So that's I don't want to say it's typical, because it's not like I found a bunch of cached prey, but it depends on if they get something big, right? If they catch a mouse, they're probably going to eat that all in one sitting. But if they get something larger, maybe they do happen to kill an adult deer. So some of these, especially larger male bobcats, could possibly take down an adult deer. And so if that's the case, they may try to cover it up with leaves and stuff so that they can come back and feed on it multiple times.
BH asks or says that they saw a dead bobcat on the side of the road in the developing area of Anne Arundel Mills near the BWI airport. And in general, would a sighting like this be of interest to anyone in case they or anybody else sees it? If we see a dead bobcat on the side of the road, unfortunately, is that something that we should report, and to whom?
Yeah, I couldn't necessarily tell you to whom. Well, I mean, I could give you names, but I don't necessarily know their numbers, or I guess I could give you email addresses. But Yeah, I mean, that's definitely something, especially that far east. Now, there had been some reports that I was sent just to kind of verify what my models were showing me as you got farther east. And so I used some of those reports that they weren't necessarily verified, right? It's just somebody calling in saying, hey, we saw a bobcat. It wasn't, there's a dead bobcat, go pick it up, or here's a photo that I got on my trail cam, here's the location. But I used some of those to look at where my occupancy model was picking up a high probability of occupancy in Washington and Frederick counties. It seemed to match up pretty well. Now, as we move farther east like that, I think that would be even more important. Because as bobcats get into those areas, how are they using areas? Where are they using areas around those high human population centers, right? Is there specific areas that they seem to be using more than others? Are they making it through those areas? Like I was saying, are they a true barrier, or are bobcats finding a way to get through those areas? So yeah, definitely report them if you can.
This goes along with what Melinda says, that she's seen bobcat tracks by a stream behind the house in Calvert County, and also found a fawn kill. I wonder if any of the like iNaturalist apps or anything like that, is that something that is collecting data that you were able to pull from in terms of correlating or utilizing in some form or fashion?
You're saying like data from the public, I guess, that I could use?
Yeah, like if they've taken a picture of it and posted it or something like that, maybe are those kind of sightings useful?
I mean, the sightings are useful, but like I said, I think I'm not sure if there's places online to just post that where it would get sent to the people who might find that really useful. As far as like the state managers, the wildlife managers, and things like that, to me, it would be smarter to just call up the DNR, and even if the person you call isn't the right person, I'm sure they can get you in contact with somebody that can get the data to the correct individual. Especially, like I said, as bobcats are moving east, figuring out where they're at, whether it be if they're just dispersing through an area, if it's more of like a travel corridor type area, or if they're setting up a home range and they're occurring in this area. I think as you get closer to Baltimore and Washington D.C., I think having that information is going to be important, just to figure out how they're utilizing those areas that humans are utilizing.
Well, Sam asks, what is pulling the bobcats to the east?
It's a good question, and I don't know if I have necessarily a definitive answer. I mean, I can speculate myself, but just based on some of the literature, I would think that you're seeing bobcat populations across the U.S. They're either stable or increasing just about everywhere. So back in the early 1900s, especially in like the 60s and 70s with the increased fur prices, maybe it's through the 80s. You saw a decline in bobcat populations, not necessarily to the point of where they were going extinct anywhere, but it was a significant decline, right? And part of it was habitat related from timber harvests over the years. There just wasn't that early successional habitat where they had a lot of prey species around. It was either I guess early on it was everything was gone, and then it started to come back, and so populations rose a bit, and then like by the time fur prices went up, I think populations were starting to decline a little bit at that point, just because the forests had matured so much. And so I think a lot of the reason is that maybe you're seeing the population that can be supported. So if you want to talk about like carrying capacity in these areas, it might be reaching that limit, right? So there's just not enough resources for bobcats in some of these areas because we have this really mature forest. There's not as many prey species, or not the abundance of prey species isn't there. And so, dispersers are having to move farther, looking for, a place where there is, some prey species to get a hold of it. And the thing is Maryland's a small area, right? You've got bobcats coming in from Pennsylvania. You've got bobcats coming in from West Virginia and Virginia. So it's hard to say where those bobcats are coming from that are popping up, on the outskirts of Baltimore. But I would assume it's partially due to that. If you're getting this population building up, this source and sink type thing, right? Where you get the source population builds up, and these individuals don't have space. There's just not as much resource, not as many resources anymore, and they've got to move to find resources.
Well, Jane asks, is anybody planning on working with West Virginia or Pennsylvania to expand the study of the bobcats that you started, to take the baton and go further?
I don't think so. Now, I can't necessarily speak for the state, but there's only so much money, right? Everything costs money. And so right now, I think the state has some other priorities they're working on. I know they're working with Seek a Deer right now. They got a project with a master's student at the University of Delaware. As far as bobcats go, though, I know West Virginia just did a bunch of different studies on bobcats. I want to say 3 or 4 different studies over the last probably, I don't know, close to 10 years maybe. Pennsylvania in the past had done a bunch of research. I believe one of the top people within the Pennsylvania Game Commission, while they were doing their graduate degrees, they did a lot of that research, and probably after. And so they've done a ton. Now, I don't know if they've done any recently, and if they're curious, about doing any more. So it's hard to say, but the resources, that's the biggest thing, is having the ability to put the money into it. I know there was a person at Frostburg who was talking about maybe trying to do another graduate project, but I don't know whatever came of that. So there's thought. I think people are interested in it. It's just whether or not the resources are there.
Jean wants to know what the sources of their mortality is. Do they get killed by cars? Is it dogs or traps or bears, or what goes on with them?
Yep. So in Maryland, there's no harvest. It's a closed season, so you can't legally harvest bobcats. Now in Pennsylvania and West Virginia, you can. Don't quote me on it, but I think it's somewhere around like 2 or 3 bobcats a piece each year in each of those states. I could be wrong. I want to say West Virginia might be more than Pennsylvania, a larger bag limit. But in Maryland, there's no harvest season, and so I mean, sure, there could be some illegal take going on. It's hard to know how many, but I'd be willing to bet that there's some illegal take, but mostly I would think bobcats getting hit by cars, or you could have bobcats killing each other, right? You could have during breeding season you get territorial bobcats, that sort of thing, or even females when they have kittens, those sorts of things. Some conflict there. Although I don't know how much that would be. I think probably the biggest thing that most people don't think about would be poison for like rats and mice. And it's not direct, right? But it's indirect. You get these small mammals eating this poison, and then and this wouldn't occur necessarily in Western Maryland as much, but if they're moving into these areas with higher human density, they're going to come across poison much more often. And so bobcats eating those small mammals that have that poison in their system, if bobcats get enough of it, it can definitely be fatal to them as well.
And then Melinda asked, is there any correlation between the increase in white deer population affecting bobcat population?
Not that I've heard of. Now, obviously, in the spring, bobcats will definitely be taking fawns, and I mentioned they can take adults, but that's somewhat rare. It's definitely possible. It's been documented, but it's not common that an adult bobcat could take down an adult white-tailed deer. So I don't know if bobcats are affecting the deer population that much. Now, I don't know if there's been any studies since that general predator survey, which was just correlating predator abundance to fawn survival. So it wasn't a direct, we've got collared fawns, and then we're following them until they die or they survive and we're getting recruited into the population. But there was a study done in Delaware recently on fawn survival, and they found that fawn survival, there's few predators in Delaware. There's really not many coyotes to speak of. There's a few in places. They're starting to see some. but they did a camera survey along with having a bunch of collared fawns, and I think they had one image out of 2 years of, I think it was like 20 or 30 cameras being run for about 2 months straight. They had one picture that may have been a coyote. It was really blurry, that the image, the animal was moving pretty fast, so it's hard to say if it was a dog or if it was a coyote. But one image over 2 years, 30 cameras for 2 months or so. And so that just goes to show there's few predators out there, and yet fawn survival was 40%. And so the bigger factors for deer, I think, are going to be weather when the fawns are being born. So if there's a lot of rain, it's a little bit cooler, they're going to get hypothermic and they're going to die. If the mothers aren't that old, they found that a lot of yearling does weren't very good mothers, and so they wouldn't feed their fawns as often, and their fawns may starve to death. But once you had adult does, 2.5 years, 3.5 years, survival was much higher. But overall, fawn survival is pretty low, even without predators. So I can't imagine that bobcats are affecting deer populations too much.
Okay. Any more questions for Kevin? He's given us a lot to think about. I'm definitely going to be not worried, not upset that I don't see a bobcat in person, but I'm going to be looking for some scat, because it seems like that's easier to find. So that will be just as exciting to me to find some bobcat scat. And I want, let's see, there's another message down here. Thank you. Yes, thank you, Kevin. This has been wonderful. And I hope that everybody comes back and joins us next week as we go tour the Beltsville Agricultural Station, virtually that is. And then we're going to look at the atmospheres of different planets as well as ours in comparison. So a lot more learning to do together here with the Natural History Society of Maryland. Stay well, stay curious, and go Bobcats with the Frostburg Bobcats. All right. Take care, everybody. Good night.
Thanks, everybody, for coming.